Bismillahirrahmanirrahim. Today we are going to discuss a paper 3 exam, which is a practical exam, but I'm going to discuss it with you theoretically so that I'm going to teach you how you're going to handle this in the lab. This is a paper which requires not only practical skills but also theoretical skills in which you can easily score 38 out of 40 in this paper. And this is the only paper in which you can score so highly because you've got to learn and you have to be smart enough to realize how to do this paper. Considering that it's a practical, but you can actually convert it into a theory paper and look at it as a theory paper, and I'm sure you can do it. This paper is for 2 hours and you have to do 2 questions on it. Now you see here, before you proceed, read carefully through the whole of question 1 and 2, plan the use of the 2 hours to make sure that you finish the whole of question 1 and the whole of question 2. So you should be spending 1 hour on one question and 1 hour on the other question. The other question is a microscope question. We won't handle that in this video. We'll do that in another video. Plant cells contain the enzyme catalase. So the enzyme is catalase. It is found in plant cells. Now, what is the substrate that we are going to use? The substrate that we are going to use is hydrogen peroxide, H2O2. So the enzyme catalase breaks the powder hydrogen peroxide and releases oxygen. So hydrogen peroxide is the substrate. Now think of it, the theory behind it. How is we are going to talking about what? So this is the substrate. When hydrogen peroxide solution, so you take hydrogen peroxide solution and then in your mind you should come, is this a 1% solution? Is this a 2% solution? Is it a 3% solution? And you are going to use samples of potato tissue, right? You're going to use pieces of potato, and these contain the enzyme. These contain the enzyme catalase, right? So when hydrogen peroxide solution and a sample of potato are put into a syringe, some of the solution comes out of the syringe nozzle as the oxygen is released. So we have a syringe and you have something, sort of a drop comes out of the nozzle of the syringe. So the drop which is going to come out is because you've got hydrogen peroxide here and you've got pieces of potato in it, and these are going to react and it's going to produce some oxygen, so there's going to be a lot of bubbles, and then a drop of this is going to come out, and you're going to count the drops in 1 minute. You are going to investigate the effect of surface area of potato tissue on the activity of catalase. So surface area is the one which you have chosen, and this will be the independent variable, which will, because we're going to investigate two things. Number one, surface area. Activity of catalase will be the dependent variable, which is going to be measured in drops per minute. How many drops comes out in 1 minute? 10 drops, 12 drops, 20 drops, 30 drops. So the drops, the number of drops will tell us what is the rate of activity of catalase. You are provided with material shown in table 1.1 and 1.2. You are provided with hydrogen peroxide is 40 cm cube and it is labeled H. Be careful to watch it. Then we have labeled P. Labeled P is the potato chips or the potato cylinders, same diameter, and they are 4 in quantity. So we would have put a petri dish, we would have labeled it P, and we would have given you 4 pieces of potato cylinders. Potato chips, just like we have potato chips when we are making. If any of H comes in contact with your skin, wash off immediately. Recommended you wear suitable eye protection and gloves to protect your hands because hydrogen peroxide is highly corrosive. So if it falls on your skin, your skin all becomes white. Before we continue with this practical, I want you to look at some diagrams and know these names of these words. One is the vernier caliper gauge. Then the other is a cork borer. We study the effect of osmosis on potatoes, so we have to cut them into these cylinders. Now, what do we use for it? What we use for it is called a cork borer, and you sort of push the piece of potato out of it, And these are different diagrams which I want you to see and have an idea how we would have prepared these in the lab and how we would have ensured that you all got the same diameter. So we used a cork borer to make these potato strips, and this is actually a cork borer, and you have another thing in it to push the pieces of potato out. So it's hollow here, and then you push this into the potato, and then you take out the cylinders. Now, part 1 of it says cut the potato cylinders in the beaker labeled P to a length of 20 millimeters. So we were asked to give you different lengths of it. So we were mostly because you see the potato size, so they were usually between 30 and 40 millimeter. So they were longer ones, more in length. So you had to cut them to 20 millimeter. Now, how would you cut them? Of course, you would keep a scale next to it, and you would measure it, and then of course you would say, okay, well, that's it, I would cut it here, and you would remove this part of it, and you would be left with a 20 millimeter you would be left with a 20 millimeter length of potato. And the diameter was fixed. The diameter was, we had been asked by Cambridge to give you a diameter of 6 to 8 millimeters. So I remember, if I say, let's say we gave you 6 millimeter diameter, so this was 6 millimeter, the diameter of it was 6 millimeter. Diameter was 6 millimeter, not the radius. The radius would be half of it. So diameter was 6 millimeter. So it says cut the potato cylinders to a length of 20 millimeter. Put the 20 millimeter cylinders back into the beaker labeled P. So you took them out, you cut them 20 millimeters, and you put the 4 pieces back again. Now, to investigate the effect of surface area, you will cut the cylinders into a different number of pieces as shown in table 1.3. Now the number of pieces were very specified. The length originally was 20, so the 4 pieces were all 20 in length. Then one was left intact. One didn't do anything, it was left intact, and it remained 20 millimeters in length. The second piece, we cut it into half. So each piece was 10 millimeter. This was also 10 millimeter, and this was also 10 millimeter. And we had 2 pieces, right? So length was 10 millimeter, but there were 2 pieces. Then the third one, we had the length was 5. So 20 divided by 4 is equal to 5. So each one was 5 millimeter, and there were 4 pieces now this time. So this was 5 millimeter, 5 millimeter, because out of the 20 we had cut it. Now, here is where the error comes, because you could not be very exact. And then the last one, we cut it into 10 pieces. So 4 pieces, 2 pieces, and 10 pieces, and each one was 2 millimeter. Each one was length of each small piece. Please read properly on the top. Length of each small piece was 2 millimeter, and these were 10 pieces. So we get this correct. Everybody needs to be very correct on this. Everybody needs to understand this, that these were 2 pieces, this was 4 pieces, and these were 10 pieces. You now need to calculate the total surface of the potato that we put into a syringe. An example how to do this is shown on page 4. On page 4, this was what was given to you. So this was all the working. One cylinder, length 20, length itna, diameter was here in this situation, this example, it is 4 millimeter. But in actual case, we are saying it was 6 millimeter. This is an example only. You have to understand that. So radius is 2 millimeter. Surface of one cylinder, 2 pi r square into 2 pi square h. H is the length. And you did this and you got this calculation. So pi is 3.14, radius of cylinder, height of length of cylinder. So this example was given to you, and from this example, you had to then calculate the total surface area. Depends on the number of pieces, n. So there was 1, then 2, and then 4, and then 10 pieces. So total surface area of one cylinder into the number of pieces. Measure the diameter of one of the cylinder and calculate. So the diameter was 6 millimeter. So the radius will be 3 millimeter. This is what was given to you. This is what I remember we gave to the students. So this was given to you. All the cylinders have the same diameter. State one other variable that should be standardized in this investigation. Now that is, of course, there are a number of things which need to be standardized, but you probably won't be able to do it now, but you can do it if you sort of read the whole question and then do the question. Then it will become easy. The different variables were volume of hydrogen peroxide, concentration of hydrogen peroxide. The temperature should have been kept constant because this is an enzyme controlled reaction, so we would have to use the temperature must be constant, and the same species of potato. You shouldn't have potato from different places and the cylinders from different, so it should be the same place, same species, same field, in fact, wherever it was grown. Then it says complete table 1.4 to calculate the total surface area of potato tissue to put in the syringe. Now show you're working in the space. Now I mean, it says again and again total surface area, but you all are confusing this and you say, Miss, why is it total surface area? Why? Because it was one piece, then there were two pieces, then there were four pieces, and then there were 10 pieces. So here you have to do what? Here is the height, millimeter. They've given you that the radius was 3 in all of them. As I told you, it was so you'll have to measure the radius, and it was the same in all of them. So that was 3 in all of them. Then you have to take surface area of one piece. Then you show you're working in this column, and then the total surface area. So here in this one, you would have to multiply. In this one, you would have to multiply by 2. In this one, you would have to multiply by 4. And in this one, you would have to, because each one is 2, so in this you'd have to multiply by 10 because each piece was 2 millimeters, so there were 10 pieces. So you would do the calculations. I will post the calculation at the end of this video, and I will do a brief, I won't do the details of it, but I will just post the answers on the end, and I will. As you finish the calculations, you'll come to a conclusion that whatever the value here, this one is going to be more than that, and this is going to be more, and then this is going to be the most. So more the pieces, more the total surface area. I keep on saying this total surface area, and that's a very important thing you understand why we are saying just not surface area, total surface area. One piece ka surface area, then two piece surface area. So it is the total surface area of first one piece, then total surface area of two pieces, then total surface area of four pieces, and then total surface area of 10 pieces. Then we come on to the next part of the question. It says, remove the plunger from a 10 centimeters cube syringe. So you take a syringe, and I'm just going to draw that. And you remove the plunger, and you put a piece of potato. So this is, I'm coloring this green. So you put a piece of potato here into it, into the empty syringe. And you remove this plunger. This plunger is this part of this is called the plunger. Some of you might not know this name. I can understand if you don't know this name. So this is the plunger. So you remove the plunger, put 120. So this is that one piece of potato, and this is 20 millimeter in length, into the barrel of the syringe, and put the plunger back. So then you put the plunger back and you push it. I'm sure you've all handled syringes and you all know what this means. So put 120 cell on the barrel of the syringe, put the plunger back into the syringe and push it to the 4 centimeter mark. Now you have to push this down to the 4 centimeter cube mark. And as you can see here, this is the 4 centimeter cube mark, as shown in figure 1.2. Now put the nozzle of the syringe into the beaker. Now you're going to put this into the beaker which is containing the shorba, which I say, or the liquid, into the beaker containing H. And now you pull the plunger up. So now you're going to suck this in, and it's going to go up, and it's going to go up. So put the nozzle in the syringe into the beaker containing H, pull the plunger up to the 10 centimeter mark. So now this plunger is, this is the potato, this is the potato, and now you've got the hydrogen peroxide all up here. But you have pulled the plunger up to the 10 centimeter. Now please don't worry about this because nobody is looking at this. You might get this wrong, you might not do it very correctly, you might take it to 10 or 9 or 8, doesn't matter, because this does not go to Cambridge. Cambridge, only your paper goes to Cambridge. So do not get nervous about it, just do it very casually. And this is all the hydrogen peroxide which is going into it. So pull the plunger to the 10 centimeter mark. Then it says hold the syringe above the beaker labeled waste. Now this beaker is waste. Waste we give you a beaker which is called waste, means that you're going to just put anything which you want to waste. Sometimes you don't have a sink next to your table. So we just give you a beaker which is labeled waste. This is for you to throw away stuff into it. So push the plunger. Now you push the plunger down. You push the plunger down so that the level of H is at the level of H is at the 5 centimeter cube mark, not the plunger, the level of H. The hydrogen peroxide is at the 5 centimeter mark as shown in figure 1.4, because some of it might come out, so that's why you're holding it onto the waste. You do not want to drop it onto the surface where you're working. Wipe the nozzle with a paper towel to remove excess H. So you wipe this place so that there's no excess H in it. Hold the syringe above the beaker labeled for waste. You keep on holding it, start timing. Now you count the number of drops released in 1 minute. So maybe it's 1 drop, 2 drop, 3 drop, 4 drop, 5 drop, 6 drop, 7 drops in 1 minute. You have to record this. You have to record the number of drops. Now whatever you have to record or whatever you have to do in the lab, the recording that you have to do, record the time or you record the number of drops. Now that is called the dependent variable. That is called the dependent variable. Right? Now put the syringe into the beaker labeled B. So you've got another beaker and you have beaker labeled B and you put that into that. Now cut a cylinder for step 1 into 2 pieces. Now you take the potato and you cut it into 2 pieces. So you cut it into 2 pieces. Each piece should be 10 millimeter in length as shown, and remove the plunger for an empty syringe. Again you do the same thing, and now you put the 2 pieces in the syringe. So you put the 2 pieces in the syringe. Now this is the syringe. I'm just going to make it again. Now you put the 2 pieces in the syringe and both the pieces are 10 millimeter in length. The original piece was 20 millimeter in length. So put the 10 millimeter in step 11 into the barrel of the syringe. Repeat step 4 to step 10. You do the same story again. Then you cut it into 4 pieces. So you take another syringe, right? And now you cut it into 4 pieces. So now you got 1, 2, 3, and 4 pieces in it. Each piece would be 5 millimeter in length, and remove the plunger, put the 4 5 millimeter pieces into the barrel of the syringe, and then you repeat step 4 to 10. Again, the same story again. And then you cut the cylinder into 10 pieces. So then you take again another syringe, remove the plunger, and now you put in the 10 pieces. So the 10 pieces you put in a syringe, remove the plunger, put the 10 pieces, and then you repeat steps 4 to 10. Now record your results in an appropriate table. So all this, even if you didn't do it, well, you could have just done it on one, and I'm not saying you do it, but I mean, you could have done it on one and you could have just cooked up the results, because actually Cambridge is not going to check any of this. They are just going to see your paper. So whether you did it or you didn't do it, it actually doesn't matter. Now, the table. Now, as you can see this table, the table always has the independent variable should always be the first column. So the number of pieces 1, 2, 4, and 10 I have added on the first column, and the number of drops, this is the dependent variable. Now I can easily I've just cooked up the results. So I said 10 drops in when 1 piece, then 12 drops in 2 pieces, and then 16 drops, number of drops in 1 minute. So number of drops in 1 minute, even if you said number of drops, it was fine. So I have made this up. I could have just not done anything and I could have just done one reading and cooked up the other results. So if you can, there's nothing Cambridge is not interested in, but basically it's the theory behind it. More the surface area, the number of drops will increase. So this is the theory behind it. And so if you had done anything which showed that, there's only one mark for that. The first mark was heading for the independent variable. The second mark was heading for the dependent variable number of drops. Then results for all the samples, whether they were right or wrong. If you gave me any results, 8, 10, 20, 30, it was fine. Then number of drops for the smallest surface area less than for the largest surface area. So we know that the 10 pieces have the largest surface area. So then that would give, but this table would only get me 4 marks because I have not specified the total surface area. So now I will add the total surface area. Now, of course, if a student did not do that, then he or she does not get, he gets the 4 out of 5. So total surface area, and this was in millimeter square. Another very important mistake that you all do is that you write millimeter in each dabba, in each box. Now this is a -1. You never do that. You always write the units in the heading. You always write the units in the heading. Please remember that. You never do that, is that you write the units in each of the little lines that you have made of the table. So you don't write millimeter in each column, and that would of course be disastrous if you did that. You don't state the independent variable as surface area. Well, I'm sorry, you won't get any marks because the answer was total surface area, and total is underlined. So if it's underlined in the mark scheme, that means it's essential marking scheme point. Then suggest an appropriate control for this investigation. You know controls are what? Either you boil the potato, which was of course the enzyme was in the potato, so boiled potato, and this is a rule that we follow, or you use distilled water instead of the hydrogen peroxide. So if you use distilled water instead of the hydrogen peroxide, you're proving that actually the hydrogen peroxide was the one which was doing all the reaction, and water mein reaction nahi hoga, so no reaction in the water. Different part of the question says identify 2 sources of error in step 3 to step 22. For each source of error, suggest an improvement. So the first error was that the drop size is variable. It's sometimes bigger, sometimes smaller. So how would you do that? How would you improve it? Is you measure the total volume dropped. Once you've counted the drops, then you collect that in a beaker, and then you use a measuring cylinder to actually measure the total volume. The second error was it was hard to cut potato pieces to equal size. So you use a vernier caliper gauge. You can see this is the vernier caliper gauge. So please, everybody should know what this is. Then it said the procedure described by step 1 to step 22 investigated the effect of surface area on catalase activity by measuring the number of drops released in 1 minute. Describe how you would modify the procedure to investigate the effect of changing temperature on the activity of catalase. There's only 1 mark, so you'll say the same surface area but use 5 different temperatures. So any 5 different temperatures, and sometimes we ask you to even be specifying them. So 20 degrees, 25 degrees, 30 degrees, 35, and 40, like this. Graph question, which is the 4-mark question, which everybody should be able to score because that's a very easy 4 marks. So they gave you a syringe and you had the potato pieces in it, and then there's a ruler, and then you were given the results are shown, percentage concentration. So this would be always, this is the independent variable, this goes on the x-axis. Now you look at the figures is 0.2 to 6, so then you spread the scale 0.2 to 6, and this of course will always go on the y-axis. So this is always going to go on the y-axis. Y-axis distance moved in 1 minute, and you have to give me the unit. So if you miss out anything and you don't write it, well, you lose a mark. And here you see the smallest figure is 11 and 73. So now you have to spread it out on the graph, and you have to draw the graph, and you're going to label the axis. You want to write this here, and the scale is the one which is specified. So there's 1 mark for labeling the axis, 1 mark for the scale, 1 for the plotting the points. Plotting the points, you either put a dot and a circle, or you put a cross. So if you use a cross, then all of them should be crosses. If you put a dot and a circle, then you put that. And then suggest an explanation for the results between 0.2% and 6% hydrogen peroxide. Now suggest an explanation for the results. More enzyme substrate complexes formed. You see, when you're increasing the hydrogen peroxide, you're increasing the substrate concentration. When you are increasing the substrate concentration, well, the time comes when the active sites are only 100. But the substrate, when it was less, well, of course, if it was only 10 and then goes up to 100, now even if you increase it to 1,000, well, there are only 100 active sites. So all active sites are saturated, and that was the answer that you were supposed to give me. Now I'm going to give you how the graph was to be done and the final the mark scheme for that, for the surface area. Now this is how you would label the axis, and this is a scale that was asked of you. So you have to check the scale. 2 centimeter was 1% and 2 centimeter was 20 millimeter on the y-axis. And then of course you plot the graphs and you plot the points and then you join them correctly. I'm just giving you an example of it. So this is the point, then this is a point. So then you correct it, you correctly join it, and then the next point, and then you join them and see that the line goes through all the points. This is one of the students, Iman, has given this to send this to me, and I'm posting that for you all. So this is the readings for the total surface area, and you would give these to me on the table as well. So thank you, Iman, and please see the calculations and understand the calculations. Similarly, you would also need to add this to the table in the beginning, which I made is the total surface area, which was of course the fifth mark for this table, because the 5 marks for this table would only be awarded to you if you had done all this story. So this would be added here once you had done that, and I've now added it to this. Now that concludes this question 1, and I will do the question 2 later on, but that of course you can't do in A1 at the moment because we haven't studied that much, but it will of course be suitable for the A2 students. Thank you once again, and I hope this is helpful, and please give me a feedback if there's anything I can improve.